So there were a couple of factors that drew me to trying to understand melanoma. My research had been focused on the fibroblasts, the cells that support tumors, and we were trying to understand what the best model system would be for trying to understand the role of fibroblasts in supporting different types of tumors. There are a couple of factors that we considered. So one was that we recognized that melanoma is a very difficult cancer to treat, that it's very aggressive, that it has a low mortality rate, that it's likely to metastasize, and that it doesn't respond well to chemotherapy. So all of those were really important to us because we wanted to have an impact on a type of tumor where we thought that we could have the biggest impact on patients. We're also motivated by the fact that it has an increasing incidence, and so that also seemed very important because melanoma would continue to be a problem for people going into the future. I was also influenced by my colleagues, Janice Mannard and Eileen White, who worked in the melanoma field, and I was very influenced by the exciting work that they were doing, and that motivated us to study melanoma. And also on top of that, there was a student in the lab named Natalie Guo, and she said she was going to take some time to think about everything that she was doing in the lab. And she said, our fibroblasts are derived from skin, and those are the fibroblasts that we've been looking at. And so she told me she thought it was a good idea to study a skin-based cancer so that the signaling would be kind of compatible between the skin-derived fibroblasts and the tumor cells themselves. And I realized that what she was saying was really perceptive. So for all of those reasons, we were drawn to study melanoma. So there are people doing amazing work in the field of melanoma research, including a lot of researchers who look at the tumor itself and are trying to find the Achilles heel of the melanoma cells. In my lab, we've been very interested in the supporting cells that surround the melanoma. So as I mentioned, we were interested in the fibroblasts, which are cells that are kind of create the structural support and create the biophysical characteristics of the tumor and the tumor microenvironment, so the things that are supporting the tumor. And so in our lab, we've been very interested in asking, what is it about the fibroblasts and the immune cells and the other cells in the tumor microenvironment that helps a tumor to grow? And so we've been trying to understand, are there things about the cells in the tumor microenvironment that make them really good at supporting tumor growth? And if we can find those things that are kind of specific to the tumor microenvironment, can we inactivate them and thereby prevent the tumor from getting the supporting structure that it needs in order to grow and thrive? And then can we take our findings from cell culture and mouse models and use those to benefit melanoma patients? So that's been one of the areas that we've been really interested in, is understanding the microenvironment, so the supporting structure for the melanoma, and trying to understand ways in which we can manipulate that in order to benefit patients. So we're incredibly grateful to the Melanoma Research Foundation. The funding from the foundation and the support from the foundation has been extremely valuable for my lab members and for me. So the experiments that we perform in the laboratory are really complicated. So we have very complicated genetically engineered mice, and these genetically engineered mice contain multiple different genes that are modified in order for us to understand the specific role of specific genes and proteins and pathways in melanoma growth. When we introduce tumors into these mice, it's a big team project and a big team effort to try to understand the impact of different pathways on the growth of the tumors. And when we collect the tumors from the mice, we try to get as much information as possible. And so we have lots of people in our lab working on the mice on those days, working really hard as a team. And we have a really terrific team in the lab working on this project. David Jelinek, Aaron Ambers, Lauren Voisin, Mitun Mitra, and Kaiser Atai have all been working on this project, along with a host of undergraduates. So the funding from the Melanoma Research Foundation has helped us to support this team, to support these complex mouse experiments, and they've even allowed us to add a new member to our team. So we're really excited that Ortiz Hunter has decided to join us, and he has a really strong background in molecular genetics, and he's been trying to understand how we can take what we found in mouse mouse models and in culture, and translate it to patients. So I would say that the benefits for kind of our lab overall of the Melanoma Research Foundation funding has been the capacity to investigate really kind of scientifically and kind of technically challenging questions, and to allow us to try to get some answers, but also to give us the opportunity to keep our teams supported, keep our mouse colonies going, and also to develop new strategies to benefit cancer patients. And for me, kind of career-wise and personally, it's been very advantageous to have the support. It's helped me to get other grants, help us to get papers published, and also for me to advance in my career. So we're incredibly grateful to the Melanoma Research Foundation. Our Melanoma Research Foundation funded research gave us an opportunity to pursue an observation that we had. So it's kind of serendipitous, but we found that if we inhibited two different pathways in the mice, that the tumors were much smaller than we'd ever seen before. So we're very grateful to the Melanoma Research Foundation for giving us an opportunity to pursue this and try to see why is it that if we inhibit these two pathways at once, the tumors become so much smaller, and is there an opportunity to build upon this finding to develop new therapies for patients. So we're very excited to have the opportunity through the Melanoma Research Foundation funding to pursue this kind of serendipitous finding that we had. More broadly, we're very interested in trying to understand how we can help patients to harness their immune system to target tumors. So for melanoma, we're actually very fortunate that there are immune checkpoint inhibitors. So that's one way that patients can, we can treat patients with melanoma, and patients with melanoma tend to respond relatively well compared to patients with tumors of other sites to immune checkpoint inhibitors. And for many patients, this has been really an important breakthrough technology. There are some limitations to immune checkpoint inhibitors. So one limitation is that not everybody responds. And the people who don't respond, there can be a lot of reasons, but at least one is that their body isn't mounting enough of an immune response so that even if you activate the T cells, which the immune checkpoint inhibitors do, there just aren't enough T cells there. And another problem with the immune checkpoint inhibitors is that there can be side effects, and that can result from the immune system being kind of too active and targeting not just the tumor, but other tissue. So we're very interested in trying to, in general, find new treatments for cancer that allow the patient to develop an immune response, but specifically at the tumor with limited immune activation elsewhere. So the Melanoma Research Foundation funding has helped us to continue to pursue this goal and allowed us to continue to come up with strategies and think about strategies that might help us to suppress a tumor by activating an immune response, make tumors that are kind of cold hotter, but also to do it in a way that will limit the amount of kind of adverse effects and systemic toxicity, so that the patients will have fewer side effects and be as healthy as possible in response to the treatment. So those are kind of our long-term goals.